Good evening, everyone. My name is Rebecca Synnott, and I'm a PhD student at the Waterford Institute of Technology. This evening, I'll be chatting to you a bit about my PhD project, which is using conservation genetics to learn more about the red squirrel populations in Ireland. I'll be mainly focusing on the red squirrels within the Ring of Gullion as part of a collaborative project with the Newry Mourne and Down District Council. Okay, so for a presentation talking all things red squirrel, I think a good place for us to start will be looking at the history of the red squirrels in Ireland and the UK. So first what we're going to look at is human impact that we've had on this species. So population declines occurred in Ireland and the UK between the 17th and 19th centuries due to both deforestation and trapping of squirrels for the fur trade. In the 19th century, the trading and sale of live red squirrels was fashionable, and up to about 20,000 animals were sold annually in London markets. These actions, along with several harsh winters that were recorded, were thought to have contributed to severe population declines of the red squirrel around Ireland. Within the 19th century, work was already being done to kind of help recover the populations. So squirrels were then imported from around southern England and continental Europe. Stock was brought in from the surrounding areas in southern England where the squirrel trading had actually occurred. This geographically diverse stock was used to restore populations around Ireland, with at least 10 documented introductions occurring over the 19th century, and we're going to have a look at these introductions just on our next slide. So as you can see looking through here, these introductions often consisted of just a few individuals, and it was their offspring that were later moved to other locations, resulted in red squirrels then being documented throughout Ireland by 1910. So as you can see, looking at some of the years that these introductions took place, kind of reintroducing these populations and then translocating their offspring around the country actually took quite a long time. So then as these numbers then started to recover, we then introduced a bit of a new problem in the form of the North American grey squirrel. So this was introduced into both Britain and Ireland in the late 19th and early 20th centuries. Its spread contributed to declines of the red squirrel in many parts due to the competitive advantage held by the grey squirrel and for competition for food, and also the introduction of disease and the role it played in the spread of squirrelpox. There's one positive thing that we can take away from that is that despite the continued presence of the grey squirrel throughout many parts of Ireland today, the most recent national squirrel survey found that the grey squirrel distribution has retracted and the species was absent from parts of its former range, although it is still invading some new areas, but the red squirrel populations are still recovering. So we do have our established red squirrel populations, but this is where my project links in. So at 10.5%, Ireland has the lowest level of forest cover in Europe after Malta. And despite extensive hedgerow networks in parts of the country, habitat fragmentation is a threat to woodland species. When this is coupled with a history of translocations, the Irish red squirrel population is likely to have high levels of risks of inbreeding and like a loss of like kind of locally adapted ecotypes. And inability for new animals to move into an area and rescue kind of the stock that had been living in that area. Such risk could ultimately lead to local extinctions. So I aim to use several genetic methods to gather information about these populations in the hope that this information can be used for conservation management decisions, such as translocations, and also looking at levels of several genetic methods. This can inform us on many different things. So we can look at the heritage of the different squirrels, we can look at the ancestral lines of where they may have come from, and we can then also look for disease, not only squirrelpox, but for diseases that might not be as noticeable through physical traits, such as adenovirus, which is a respiratory disease. I can look at the levels of inbreeding and outbreeding within populations, which will help provide information for future translocations. you obviously don't want to move any squirrels that might be potentially sick and infect a new population. Having looked at levels of inbreedings within population to see do they potentially need some new squirrels introduced to help keep good levels of genetic diversity, but also you don't want to introduce someone who is too genetically different, and this might cause a thing called outbreeding, such as maybe reducing resistance to disease. I'll be using a couple different methods that can provide different information. So as I said, we want to kind of look at the ancestry of the squirrels. And for that, you would use something called mitochondrial DNA, or it's shortened to mtDNA. Sometimes you might, if you listen to other talks, you might hear people refer to things such as haplotypes. So this is a long sequence of DNA, and it is slower to evolve. It retains genetic footprints and historical movements and migration patterns. So I could compare this to maybe squirrel DNA in the UK or around Europe to see do they have the same ancestry lines. And then there's also a thing called nuclear DNA. So this will provide a more up-to-date kind of insight into population dynamics. You can use this to look at individuals or to look at genetic diversity within a group. So far what I've talked about is all very broad in relation to squirrels all around Ireland. But specifically for the rest of this talk, I will be focusing on the red squirrels in the Ring of Gullion. I started a study up around the Ring of Gullion in March of this year. So what I want to do is I want to apply these different genetic methods to learn more about these populations. The Ring of Gullion has quite a fragmented forest system, so I want to have a look at how that may have affected the squirrels in this area. So the main points that I am hoping to get some information for is to look at the local information on the genetic makeup of red squirrel populations in the forest of Sleep Gullion. This information can be used for future translocations if needed, not just around Sleep Gullion, but with squirrels all around Northern Ireland, but also all around the Republic as well. I'd also like to learn a bit more about population density estimates of red squirrels within different locations, and to do a comparison of methods to see what might be the most cost-effective but still really accurate method for recording red squirrel populations. So I'm doing a comparison between camera trapping, live trap and release, and also some non-invasive methods using hair tubes. What I'm hoping to get from this is then by testing if you could just use a camera and a feeder setup to then get an accurate means of a density estimate, which could be used as a more simple way to monitor populations within the future. So I have sampled at a few sites around Sleep Gullion, and I'm just going to show you a few of them here. So I mainly focused around Sleep Gullion itself. So I have a site called here Clevey, Old Growth, which is what I've named an area just above the playground and the forest drive. Longfield, Squirrel Hide, which is down near the gun club for anyone who's familiar with the area, and a site just down along the forest drive as well. I've also gone a bit further out to have a look at the squirrels in Camla as well. Obviously these populations will be quite separated from each other, so I'd like to see if I can see many differences between them, or if there is any way that the squirrels have possibly been able to travel to promote some gene flow. So I just want to go through some of the methods that are being used within this survey. So I've been using some camera and feeder setups. The bait that I use for the feeders is a 50-50 mix between peanuts and sunflower seeds. This is great. It's a really accessible method, and it's one that volunteers can get involved in with as well for any future surveys that are being done. If we need some more information in the future about some of these populations. This method is great for confirming kind of presence or absence of red squirrels within an area, and can also give you an idea of squirrel health if you can spot if there maybe are squirrel pox in the area, if you can then see some lesions on some squirrels in any photos. Another method that I was doing during the survey was trap and release. So the traps are always checked quite regularly. I would open the traps at dawn in the morning, and then I would go back and I would check them mid-morning and then in the afternoon, and then the traps would get closed in the evening, so there was never any chance of squirrels getting trapped in overnight, and at most they were only ever in a trap for a couple of hours. They were provided with food in the trap, and also, as you can see, I have tarpaulin around the trap here as well, so it was protected from any weather conditions. Once I did go and check the traps and check the squirrels, I would take note of things such as sex, and then I would also, with squirrels, just kind of check them generally. I would take a little bit of hair just from the squirrel, and that is what I'm going to use for my genetic study. So it's all very non-invasive, nothing harsh is done to squirrels, and then they are released to run off on their merry way. Okay, and then so when I was doing the live trapping for these squirrels, I would follow this very simple kind of sampling design. So as you can see, these blue circles here would represent the grid in which the traps would form. So they're all separated by about 30 meters. So to do this trapping, you'd need an area that was roughly 120 meters by 60. I would set up 10 traps at a time, and at most I would only ever have 20 traps open in two sites that are very close to each other, so I could check them all in a timely manner. This sampling design allowed for trapping within a site, and then allowing for a lot of recaptures of the same squirrels, so that we could then produce density estimates for that particular area that we're looking at. These little dots over here in the white are grid locations that I set up traps at one of the sites, which was near the gun club, the one that I referred to as Squirrel Hide. These yellow squares here refer to my non-invasive sampling method, which is hair tubes, which I'll be chatting to a bit about next. So for the hair tubes, it's a really simple, like accessible way of surveying animals, and it's, as I said, very non-invasive and very non-disruptive to the animals. So essentially, all it consists of is a small bit of PVC pipe that you secure to a tree. And then I create little kind of sticky hair patches that go on the top of the pipe, so that when the squirrel runs in to get the bait that's at the other end, a little bit of hair will just stick to the patch, and then the squirrel will kind of run on out, none the wiser. This is also quite a nice sampling method, as you then just go and you check the tubes once a week to refill them with bait, and then you just collect your hair patch and you just insert a new one into the top of the PVC pipe. This will provide hair samples that can be used then for those further genetic methods that I was talking about. So even though you won't just get to see the animal, you'll then actually get a lot of information about them just from this little bit of hair. I'll be able to distinguish individuals, be able to learn a little bit about the diversity within the population and how many individuals that have been visiting the tubes. The sampling design for the hair tubes is in a similar kind of grid fashion as the live trapping. As you can see here, with these yellow squares, we have kind of 3 lines within our grid. And as you can see, these are the ones that I plotted at a site where I actually did some hair tube work. In a similar fashion, we're hoping that by having, I guess, the increased amount of hair tubes, that we can also then get recaptures of the same individuals visiting the same tubes. So that can also then be used to produce density estimates for squirrels in this area. The genetics work for this is still ongoing at the moment, but I have been able to get some information from the sites of where our trap and release methods have occurred based on numbers that we had there. So I have rough ideas of how many individuals are within some of the sites, and we can also look at the sex ratios, so how many males and females we have in each area. It's quite interesting, as we can see, that the numbers are actually fairly even in the 3 that I have so far. So within the 7 individuals within the Kalibi site, there are 3 males and 4 females. Squirrel hide actually had quite a large amount of individuals, larger than is expected. So we have 7 males here and 6 females, and then Longfield is a 50-50 split of males and females. So what would be interesting now to see with the numbers that we have here is to actually then see how closely related these individuals are to each other within each of the sites. So the great thing about the kind of live trap and release is that yes, you can get like these instant numbers. So then that brings us to our hair samples. So they're currently being worked on, but so this is how they're going to be used and the types of information that they can then provide us. So as you can see here in this tube, I just have a few strands of hair. That's going to break down, and then I'm going to extract DNA from that. This is a sample that I have already done. The thing about the hair, though, is that you don't know what species it has come from. So in all the areas that I sampled, thankfully I did not see any grey squirrels, so it was only red squirrels that I came across on my cameras or through trapping. But there are also pine marten in the area, and the pine marten are quite fond of reaching in and getting some of the bait for the squirrels. So what you then do is you perform a species ID test. So then you can then identify which species that the hair has come from. And then you'll take on your red squirrel samples, and you can then identify individuals. You can look at how genetically diverse then the population might be, and then using that mitochondrial DNA that I spoke about, identify different ancestry lines and look at where these squirrels may have come from, where they may have moved around the country in previous translocations, and then hopefully use this information to identify where they can be moved to if it's necessary. Basically, what I'm hoping to do is just provide as much information as possible to inform future conservation and management decisions that may be needed for the species. And the more information that we can gather about them, the better. So as I said previously, this work only began in March of this year. So a lot of this is still being processed and results are still currently being generated. So I'm hoping to have some very interesting information for you all in the near future. This work would not have been possible without all these wonderful people. I want to say thank you to them all for all the ways that they have helped me. And I want to thank you all for listening. I really appreciate your time. And here is my contact information if anyone would like to learn any more about this project, or if there are ways to get involved with, say, doing some non-invasive surveys, such as the hair tubes, and how to get involved with that. That would be amazing. If you've any other questions about anything Red Squirrel, please feel free to contact me. Thank you.